Hello everyone, good afternoon. It's Gautam. So today's session we are going to discuss about the cell, the unit of life. In the last session, we have completed about the prokaryotic cell, the outside extensions, cell membrane extensions of the cell, prokaryotic cell or bacteria. We have discussed in the last session. So in today's session, we are going to discuss the continuation of that. So we'll see. Let's start the session. So we have discussed cell envelope of bacteria in the last session. Cell envelope in the sense we have discussed what are the envelope, what are the layers which are present or covered the bacteria there. So we have discussed. So let me recall that with a quick recap. So here a cell membrane will be there. Upon that it is followed by there is cell wall will be there, and the outermost layer, that is glycocalyx, will be there. So three together it will be working as a single protective unit. The cell membrane, it functions as a selectively permeable membrane. So if a cell is there like this bacterial cell, so if you take look out of this one, the cell envelope there, so you will get three layers. The innermost layer is cell membrane, next to that cell wall, next to that glycocalyx will be there, and each, so three of them together they will be working as single protective unit. But if you see a single one by one, their functions is distinct, their functions is different. There are some differences are there in each layer there. So if you check here glycocalyx, it is of two types you will be seeing. If it is thick and tough, then we are calling this as capsule. If it is having thin and like loose sheath, then we are calling it as slime layer. And the glycocalyx function is it will be producing some sticky nature where it can attach us to the cell, the host or uh, the substratum or rock or in a stream, it will be attaching to the substratum there. So or within our body, it can attach us, like you can take dental plaques, the dental plaques where a yellow layer, the dental plaque is there, no? That plaques or plaques, it is due to this bacterial attachment there. So sticky nature it will be producing, and if you see the cell wall it will be giving a shape and it will be protecting the cell from the cell bursting or collapsing. If bacterial cell has entered my body, then it will be uh, so our body will be producing some defense mechanism to fight against that foreign organism there or the pathogen of bacteria there. So that bacteria will be protect themselves from this cell wall, which is having the protective unit. It is a protective unit, and it also prevent from the bursting or cell collapsing there. So to avoid this one, to kill the bacteria which is having this cell wall, so we will be taking some antibiotics there or like penicillin, which destroys the cell wall of the bacteria. So that we have discussed in the last section there. And the third one, the innermost one is cell membrane, which is having uh, the cell membrane, which is having selectively permeable function, and which connects to the outside world. So it is about cell envelope. In the last class we have discussed about this. Cell wall composition, when it comes to the cell wall of bacteria composition, it is made up of peptidoglycan. We have seen in the last section. The peptidoglycan, peptido word is there, no? It refers to the protein, glycan refers to the polysaccharide. So together it is making a code that is composition of cell wall in bacterial case. And uh, in archaebacteria means the bacterial ancestors. which came first, the ancient bacteria, the ancestor of these bacteria. So when origin of Earth was taken place, there the archaebacteria were existent. So the archaebacteria can live in extreme conditions like in hot volcano or hot springs or even in extreme saline conditions and all. So that we will learn in different chapter. So there the cell wall is made up of pseudomurine. There the cell wall is made up of pseudomurine. That is the composition of bacteria cell wall. And we have seen gram positive and gram negative bacteria based on the staining procedure. So which was given by Christian Gram, and the gram which is the stain which is used is crystal violet stain is used. The bacteria which retains the stain, we call them as gram positive bacteria. The bacteria which does not retain the stain, we call them as gram negative bacteria. So in the last session we have discussed about this, and we can solve these questions also. The term glycocalyx is used for a layer surrounding the cell wall of bacteria. So yesterday we have discussed, I think, this one. So last session we have discussed all these questions. So we'll see the content. Mesosomes also completed in the last session. So we'll take a quick recap of this one also. Mesosome. Mesosome is nothing but the infoldings of this cell membrane there. The infolding of this cell membrane, we call this as a mesosome, or the extensions towards the cytoplasm. See, if a cell is like this, the cell membrane extension will be taking place towards the cytoplasm here. This is cytoplasm. So inside the bacterial cell, the cytoplasm will be there, and the extensions will be towards cytoplasm. So those bodies we are calling it as mesosomes. So these mesosomes got many functions there. Five functions we have discussed yesterday. So these mesosomes also we can refer it as like mini power stations. Mini power stations in the sense, so it is working like a where energy can be produced there. Like in eukaryotic cell, in cells of our body, mitochondria are there. Mitochondria which are energy, so we call them as a powerhouse of the cell. Means which is where the cell is extracting energy. The energy production will be taking place. So in case of bacterial cells, these mesosomes are there. So in case of bacterial cells, so the mini power stations we call it as, because mesosomes also will be helping in them for the energy production there. So mesosomes will be there. So the functions we have discussed there, the cell wall formation, and DNA replication or duplication, DNA replication, and the distribution in daughter cells. So it is also performed by mesosomes there, and increasing in surface area, and enzymatic content, and it also helps in respiration. So that's why we are calling it as mini power station there. It also helps in respiration there. So these are the functions which we have discussed in yesterday's class, and these are the functions of mesosomes in case of prokaryotic cell. So other than this, what are the any other extensions are there? Means that extensions are there, the cell surface of the appendages we call that as, and before that we'll discuss here only the infoldings. Apart from this, other than these mesosomes, so it is vesicle, it is tubule, and it is lamellae we call it as. So other than this, some extensions are there where it will be extended. So where some extensions are, which they contains pigments upon this. So some extensions are there where we can see the pigments like this. We can see the pigments. Pigments are nothing but the coloring substances which are present there. And these pigments which are present on this infolding of this cell membrane, this we call this as, this one we call this as chromatophore. This one we call this as chromatophore. Chromatophore in the sense, see, chromatophore in the sense, in the name itself is the chroma, that is coloring body, and phore is nothing but the coloring body which is present in prokaryotic cells. So here chromatophore of this bacterial cells, so which has pigments upon this, and of course what is the function of pigments? So it will absorb some sunlight radiations of different wavelengths and it will emit. So if you want, if you want me to simplify, take green leaf, green color it is. Why it is appear green color there? It reflects a green color. So means it absorbs all the light radiations except green. So that's why the leaf it appears to be green. So if it is appearing black, it is absorbing all light radiations of different wavelengths. So that's why it is absorbing all the light radiation. So there is no emitting of any color there. So it is, that's why it is black here. So when you take green leaf, so it absorbs all the sunlight radiations except green there, and it reflects the green light, isn't it? So here in case of chromatophore, it will be having some pigments and it absorbs some light radiations of different wavelengths. So after absorbing, what happens? So after absorbing, it will emit some light radiation, and in case of leaf we know that there will be chloroplast will be there. So there is a chloroplast is there. So function of chloroplast, what is the function of chloroplast here? Chloroplast, it has chlorophyll pigment, and a chlorophyll pigment means which absorbs all the wavelength radiations except green there, and this pigment it will be absorbing like blue to red, blue and red wavelength of spectrum there, so that light radiation is absorbed, and this chloroplast involved in photosynthesis there. So we know that the green color leaf which contains chloroplast, it absorbs with the help of pigment, it absorbs the light radiations and it performs photosynthesis there. So in the same case, in case of this, in case of bacteria, so earlier, just imagine when earth was originated, there were no plants, there were no animals, there were only microorganisms were existing at the time, and before that there were no microorganisms at all. So I am talking about the earth condition where only bacteria are existing. So in such cases, there is no plants also. So there need, in atmosphere there is no oxygen also. But initially how the oxygen is formed is with the help of this bacteria. That bacteria which is having chromatophore is, that is cyanobacteria. That is cyanobacteria. So cyanobacteria is chromatophore, and chromatophore is helping what? It is absorbing light radiations with the help of these pigments, and with the help of these pigments, when it absorbs the light radiation, it performs synthesis of food there. So this chromatophore helps in photosynthesis. Means without the chloroplast performing photosynthesis here, that is food for the bacteria. So synthesis of food by using light radiation, and these are the organisms. Cyanobacteria are nothing but here cyanobacteria is given. So cyanobacteria are nothing but these bacteria which produced or released oxygen, evolution of oxygen in the earlier time when the earth was just a microorganism planet at the time. So this oxygen was evolved by these cyanobacteria, and this is the chromatophore. This one is chromatophore here. This is chromatophore, and it is also enfolding. It is also enfolding, but with the pigments. It contains pigments here. So these are pigments. Okay, it is about the enfoldings towards the cytoplasm of the cell membrane of mesosome, and how they are given. If you see in the uh, NCRT lines, you can get all this information. So it is about the cyanobacteria, and next we will be seeing about the cell surface appendages. Cell surface appendages in the sense, see, I am drawing a bacteria now. So this bacteria will be having some cell surfaces like this. So which is, so it will be having flagella. So it will be having flagella, and it will be having many. So it will be having many bristle-like structures like this. This is cell surface which has many bristle-like bristle-shaped structure, and that we call this as fimbriae. And apart from this, even you can see there are some pili structures which are somewhat lengthy in structure. So these are pili or pilus. So that is pilus or pili. So that these three are the cell surface appendages. So let me explain this in depth. So now what we are seeing is cell surface appendages here. So this one is flagella, and this is pili or pilus. So it is singular and plural word there, and this one is fimbriae. So it is fimbriae. So if you see here, in case of flagella, so one by one we will going to be study here. So if you see the flagella, that is the extension of this cell membrane. It is the extension of cell membrane there, and the extension means it is not towards in cytoplasm inner side there. It is outward extension, and in NCRT they have given flagella is the structure which came means which is from cell wall. They have given from cell wall is given, but if it is from cell wall means, see, the cell wall will be there here. This is cell wall. So surrounding to that you will be seeing cell wall. The flagella is a structure which is coming from the cell wall, but it is not originating from the cell wall. So it is originating, it is the membrane, that is the plasma membrane. So this layer is continuation of plasma membrane, not the cell wall there. But in NCRT lines, so let me show that so that you will not get confused. Not only in NCRT lines, even in your modules they have given like this. The cell flagella is the structure. When it opens, I will show that. Please. So this flagella is having the three parts will be there. So that three parts are the flagellal three parts is. So the long slender like structure is there. No? This long slender like structure. So this long slender like structure we call this as filament. So the long slender structure of bacterial flagella is filament, and this part, we call this as basal body. We call this as basal body. So from here to here, this is basal body, and next to that, what you are seeing in this region, this is hook. This is hook here. So from here to here, it is a hook. So these three parts, the flagella has of three parts there. That is, one is basal body, and this is hook, and the long slender hair-like filament, that is hair-like structure, that is filament. So these are the three parts of flagella, and where it is used, the function of flagella here is function of flagella is, so that is for motility, for the movement, or for movement it is used. Motility or movement, both are same here. Motility or the movement, both are same here. And the bacteria cannot walk like us. The bacteria cannot walk like this. So they will be using this flagella, they will be showing movement. So they will swim, they will spin, they will spin in the stream, they will swim in the blood. So they will swim in the water stream. So they will be using, with the help of this flagella filament, that is the flagella, it will be used for swimming purpose. The swimming or spinning, it will be used for the movement or the motility, it is used. And if you check the pilus here, next is pilus, and it is made up of some filaments there, some proteins are used here. If you cut, see, it is a flagella, no? If you cut this one, it is a round shape, it will be having. So if you check this pen, it is having some inside of this one, like you feel like it will be there, no? So the same kind, it is made up of many protein filaments there. So here, if you see here, so many protein elements you will be seeing. So many protein elements will be there. When you cut that one, you can see the protein elements there. So that you will be seeing, that in depth we will be studying about the structure of flagella and cilia in the same chapter only. So these are the proteinaceous filaments we can see in the proteinaceous structure which will be present in this flagella there. So that it is made up of flagellin protein there. So when it comes to this pilus, here next is pilus. So the same pilus here. This pilus, it is made up of a special protein there, that is pillin protein is. So pillin is the protein. So pillin is the protein which is present in this pilus, and the function, when it comes to the function of this pilus, so these bacteria will be having all these appendages. That is, when it comes to the function of pilus here, that is, it is used in attaching or adhering. Both are same, attaching or adhering to the host, tissue. Host tissue in the sense, see, some bacteria is there in body, in my body, the bacteria is there. The bacteria will be floating, no? The bacteria will be moving. So when I am eating the food, I am taking the food along with the bacteria. That bacteria has entered my gut now, food pipe, stomach, and it follows the small intestine and all. So it has to attach to the some walls of intestine or walls of food pipe there. So if food pipe is there here, inside of this one, so it has to attach. So the attaching function, it will be done by this pillin there. So even pillin is also used for holding or stinging, attaching, adhering to the host tissue, or not only host tissue, even not only in host tissue, even it is in stream. So in stream, you can see in the stream also, like rock or substratum in the stream. Rock in stream or substratum, both are same. So rock in the stream, that is, it is attaching, and it has got another function also, which it will be working as a sex pilus or sex pili. Sex pili in the sense, see, it will be used in the conjugation process. Conjugation in the sense, conjugation in the sense, the bacteria is there here, one bacteria is there, and another bacteria is there. So when they are actually, if you see in bacterial case, there is no sexual reproduction is there. No sexual reproduction will be taking place. But a fond of, a kind of sexual reproduction is seen in case of bacteria. Means to exchange the genetic material or to exchange the DNA, this pilus will connect with the another bacteria there. So this pilus, it will connect with the another bacteria, through where this bacteria DNA will be transferring like this. So that we call it as, that process we call this as conjugation. So that is nothing but conjugation. So the conjugation is nothing but the bacterial DNA transfer from one bacterial to another bacterial DNA. So it is helping in the way of sexual reproduction to create variations there. This is used. So in sexual reproduction, to kind of sexual reproduction where variations are seen by transferring DNA through pilus here. This is pilus. So it is pilus here. Or pili. So this is the function of pilus or pili, the extracellular appendages what you are seeing, that is the pilus there. And next is fimbriae. So the function of fimbriae is also same somewhat. So if you see the fimbriae here, so this structure, it is like Bristol shaped structure. Bristol shaped in the sense, you might have, like you just see the brush, toothbrush. It is having bristles, no? So like that, many bristles will be there here. So many Bristol-like structures. So the difference between fimbriae and pili is, pilus or pili is somewhat long and they are less in number compared to the fimbriae. Fimbriae or fimbriae or fimbriae are less, more in number and short in length, and it is Bristol shaped structure you see here. And the coming to the function, it is also used for attaching the. So when it comes to the function, it is also used for attaching or adhering to the host tissue or rock in the stream case. So it is used. That is fimbriae. It is about the cell surface appendages. And now I want to discuss about the plasmid. Plasmid also. So where we will be seeing that within the bacterial cell. So that is plasmid. So the topic is plasmid here. So in the last class, I was just gave introduction of this one, but in depth we need to discuss. See, these plasmids are nothing but what? So here it is a bacterial cell, it is a prokaryotic cell, and it will be having a genetic material there, where it will be laid in the nucleoid region. This is the genetic material. So apart from this genetic material, you will be seeing some special structures which are extra chromosomal circular DNA, what we are calling it. Extra chromosomal circular double stranded DNA is nothing but plasmid. So extra chromosomal means what? It is the main genetic material. Main genetic material is there already. Main genetic material is nothing but the main chromosome. The main DNA is there in this bacteria. But apart from this, these are extra chromosomal. So these are, the plasmids are what? These are extra chromosomal double stranded circular DNA. So these are extra chromosomal double stranded circular DNA. We call this as plasmids here. So coming to the functions of this, where do we see? So in bacterial, in bacteria case, we will be seeing that plasmid. So this special thing about this, you can take an analogy of this one, where plasmids, it is like a extra software installed in this. So in laptop, what will be there? So in laptop, there will be some system apps will be there. But to protect some this software of this uh, laptop, we will be installing some malicious antiviruses there. So to prevent the virus attacks or malicious software, malicious apps or malicious that viruses. So that to prevent that, we will install some antivirus software in it. So it is like this one. So the plasmids are having some extra software installed in it, or it is like a pen drive which is carrying some extra software. Where you will be seeing in some bacteria it will be there, in some bacteria it won't be there, and the bacteria which has these features, they will survive. So example for this, you can take example. So when we get infected with the bacteria, we will take what? We will take antibiotic resistance, means antibiotic tablets we will take. When we take antibiotic tablets, or don't take off antibiotics, see, so we have inhaled bacteria here. So we have inhaled, bacteria, and it is going to affect our lungs here. So it is going to affect our lungs. So the bacteria is inhaled and it is attached somewhere in this part of trachea. That air pipe is there, no? Trachea, it is attached somewhere by using that villi and all. So the cell surface appendages, it is attached here. Whatever blood cells or what our body will do. So now our body will get to know that there is some foreign organism there, causative organism, pathogen is there, bacteria has entered body. So then our body will be producing some, like WBC cells are producing, and they will come and trying to defend, throw it out, means to fight against this bacteria. So this bacteria, what it will do? It will it will also fight against this one. It will be having some protective outer coats and all. So now it will not, these WBC cells, natural immunity of our body cannot fight with these bacteria, and our body cells will give up. So when our body cells will give up, so then we'll go for the treatment, and we'll go for the medical, or we'll go for the doctor prescription, and they will give some antibiotic tablets there. So when we take antibiotics, antibiotic tablet. So it will also come and fight against these bacteria. The bacteria will resist that dependent, means the antibiotic tablet which we have taken, it will trying to fight with this bacteria, no, but this bacteria will defend it, means it will resist it, it will stop that, it will hold that, it can resist that antibiotic chemical which is coming and fighting with these bacteria. So that is nothing but antibiotic resistance. So that antibiotic resistance is expressed by these plasmid genes there. So these plasmids are nothing but these plasmids are nothing but what? So plasmids are nothing but these are extra installed software. So which confers some phenotypic characters here. So which confers some phenotypic characters. Which means, see, if bacteria want to survive in my body, it has to produce some resistance against the that antibodies which are created in my body. So in that case, it is protecting themselves, no, that is, it confers some phenotypic characters. How it will be having? It will be preserving its own phenotypic character. So by antibiotic resistance genes there. By providing or by producing antibiotic resistance. And one more case we will be using this plasmid. So this is in case of this bacteria which is using for themselves. So bacteria, these are having plasmids for themselves to protect against the other organism or other chemicals there, and it will be showing some releasing some, it will be having some genes, it will be having some genes which will be producing antibiotic resistance there. So that antibiotic resistance will be produced by them. And the plasmid is also used in human services. Human services in the case, like we will be using the plasmids of these bacteria for ourselves. So that using of bacteria for ourselves in humankind services in case of biotechnology. In biotechnology, so we will be using this plasmid in case of recombinant biotechnology or rDNA technology. So how we will use in the sense, see, if I want to produce the insulin gene, so in my body I am having enough insulin producing genes, and the insulin is producing in a normal condition. But some persons, their diabetic conditions where insulin will be high and low, it will be there. So in that cases, if they want to take insulin from my body and to give that person, so then they will, what they will do, so they will take a gene which is responsible to produce the protein, that is sorry, that hormone, that is insulin hormone. See, insulin is there, so I will simplify this one. In my body there is a DNA is there, and the DNA which expresses here, so the DNA, this part of DNA, this part of DNA which express in the sense, so say, listen here, DNA is there, and the DNA stores some information to prepare insulin. It has some information to prepare insulin, so it will make RNA, and RNA will translate this code which is present here to make protein, that is insulin is prepared. So this is how it is prepared. So this gene is responsible. Now what they will do is, in this case of biotechnology, plasmid rDNA technology, they will take this plasmid, they will take this plasmid here, so they will take the plasmid and they will cut the, so they will cut this plasmid here, and they will insert they will insert this part of gene which is responsible for insulin. Here they will insert. So they inserted the human gene in the bacterial plasmid, and this bacterial plasmid is there, no? Again they will introduce into the bacterial cell. So they will introduce in the bacterial cell again. So when this plasmid is introduced in the bacterial cell, so they will replicate. So they will replicate in the sense the another plasmid is formed. So the bacteria does not know that it is a foreign DNA. Bacteria will think that it is of my own DNA. Then it will replicate the same one. Replica means creating the same copy of this plasmid. So it is creating the same copy. So when it is creating the same copy along with this plasmid, I am getting the copy of this insulin gene also. So this is how insulin is produced in a huge content in so it will be used in rDNA technology here. So here rDNA technology or biotechnology, it will be using that in plasmid in case of human services like this. So they will be taking the desired gene of interest to incorporate in this plasmid. They will cut the plasmid, original plasmid they will cut, and they will insert our gene of interest, and they will again introduce to the bacterial cell. So when they introduce, the plasmids will replicate the creating the same copies, and it will be resulting in the doubling our gene of interest also. So this is how it is. I am just giving one example where plasmid is used in case of biotechnology, recombinant DNA technology. It is also used as recombinant DNA technology in the recombinant DNA technology. So it is about that. So okay, coming to here in the NCERT cell, I said no. Coming here, see here, this I wanted to show here. In case of bacterial cells, the motile or non-motile, if motile, they have thin filamentous extension. So that is from their cell wall is given. See, you need to note here from their cell wall. From their cell wall. It is here in the sense, see, in the last case, I said that this flagella, it is extension from the cell membrane. It is extending from the cell membrane, but from their cell wall. So they have thin filamentous extension from their cell wall called flagella, and it is having proteins and all. So you read that later. So it is about the flagella. And plasmid word also. You just check here in the NCERT line. Just check here in the NCERT lines where they mentioned about the plasmid. So these words initially we don't get understand. So you need to watch this, revise this properly. So here they have given the smaller DNA are called plasmid. The extra chromosomal small circular outside genomic DNA, we are calling this as plasmid. This plasmid DNA confers certain unique phenotypic characters to such bacteria. I said no. So it is having some unique phenotypic character here. Unique phenotypic character in the sense it can resist. So one such character is resistance to antibiotics. So one such character is resistance to antibiotics. That is one such character. You will get many other characters which are conferred, which contains by this plasmid also. So in higher classes, that in the 12th standard we will be learning about the plasmid DNA used to monitor the bacterial transformation with the foreign DNA. So this is foreign DNA. So my insulin gene is there, no? That gene of interest is a foreign DNA. So that foreign DNA is incorporated into this plasmid. So monitor bacterial transformation with the foreign DNA And nuclear membrane is found. Okay, it is about this plasmid. Only this line is there. So in biotechnology, we will be using that to monitor the foreign DNA, to monitor transformation of foreign DNA. This I am incorporating from, this is human DNA, no? So this is human DNA. So from human DNA, I am taking insulin gene here. I am taking insulin gene and I am incorporating in this. After this, I am introducing that into the bacterial cell. So the bacterial cell, it will replicate the insulin gene. So as it doubles, so we'll get this one. So it is about the plasmid. Before going to this ribosome and inclusion bodies, if we have any questions, we'll discuss here. Okay, principal protein of cilia and flagella is. So there is regarding this means in depth of the flagella structure and cilia structure, we will going to deal in the next session. But for you, I will give answer for this one, that is tubulin. Tubulin proteins will be there within that, which helps in formation of cilia and flagella. Next, motile bacteria are able to move by villi, fimbriae, flagella and cilia is given. So don't get confused here. Motile bacteria are able to move by what are the organ is or the part is, that is flagella, not fimbriae and villi. So they are very small. In case of pilus or villi, it will use just to pull like this, holding and pulling like this one, that is villi or pilus. But in case of flagella, it is not. By rotating the flagella like this, by it will be using as a rotor and it will swim. So flagella is the answer here. Structure that helps some bacteria to attach rocks and host tissues. So if you see the fimbriae, mesosome, holdfast and rhizoid is given. So these are not answers. So we will see in some different chapter there. Mesosome also. Mesosome is extension towards cytoplasm. So the fimbriae, only fimbriae is the answer here. Structure that helps some bacteria to attach rock and host tissue. And regarding this question, we will discuss that later in this ribosome case. So I will just give an short intro of ribosome so that we will continue in the next class. So ribosomes are nothing but what? So in general, ribosomes are nothing but what? So just think, ribosomes means, ribosomes are nothing but these are protein factories which can make proteins. And the special thing about ribosomes here is, the special thing about ribosomes is that there is no membrane bound organelle. It don't have any membrane. So it is no, there's no membrane. And even in inclusion bodies also, even in inclusion bodies, it is also having no membrane. No membrane is present here. Even here also no membrane, there also no membrane. But the function is different. Here protein synthesis. So it is also called as pallid particles. Why they named it is pallid particles? Because of the scientist George Pallid. And regarding the structure of ribosome and function of ribosome, structure of inclusion body and the function of inclusion body, that we will discuss in the next session. So till that, bye, take care.